Hi everyone, this is part one of urinary system. So your kidneys are a major excretory organ. They help to maintain the internal environment by regulating the total water volume and the total solute, meaning salt mostly, but potassium as well, concentration in that water. They regulate the ion concentration in the extracellular fluid, make sure that you keep your acid-base balance, of course excrete metabolic waste toxins and drugs, and they also produce a hormone called erythropoietin, which helps to regulate the red blood cell production, and it also produces a hormone called renin that regulates your blood pressure. And I know it says the opposite of that on the slide, but you need to flip those two. I keep trying to remember to do that. I keep forgetting. So erythropoietin has to do with blood cells. Just think erythrocyte. And renin, as we've already talked about a couple of other times, has to do with the renin aldosterone angiotensin system. Some other things that your kidneys do is activate vitamin D and then finally carry out gluconeogenesis that we talked about in the last chapter. As a reminder, gluconeogenesis is the formation of glucose from a non-carbohydrate source. So we have the kidneys, which are part of the urinary system. Then each kidney has a ureter. There are people who have more than one ureter. It doesn't change anything or affect anything. So the ureters are taking the urine that is formed by the kidneys to the urinary bladder. The bladder is just a temporary storage facility until you can void that urine, and then finally the urethra, which carries it to the outside world. So taking a look at all of the structures, this is where they are located. And then looking at them in a cadaver. So our kidneys, we're looking at the gross anatomy now. Our kidneys are referred to as being retroperitoneal. They are in the upper lumbar region of your back, located between T12 and L5. The right kidney is a little bit lower than the left kidney because of the liver. And then sitting on top of each kidney, like two little hats, are the suprarenal or adrenal glands. The convex surface or the rounded surface is on the outside or the lateral side, and then you have a little indention on the medial aspect of each kidney. That concave medial surface is what they call the renal hilum, and that is leading into the internal space called the renal sinus. And through there, the ureters, the renal blood vessels, lymphatics, and nerves enter and exit. So showing you the location from behind, and you notice that the right kidney is a little bit lower. There's three layers of supported tissue that are surrounding the kidney. So remember, it is not part of that big bag that all of our guts are in. It is actually sitting back behind it. So they're actually embedded in the musculature of your lower back. So the renal fascia is what anchors the dense fibrous connective tissue, anchors the kidney into place. And then you've got a fat cushion around it called the perirenal fat capsule. And then you have the fibrous capsule, which is on the outside of the kidney. It's transparent. On our models, it's kind of a dark maroony color, and that helps to prevent any spread of infection to the kidney. So looking at this again, the top is the front and the back. You can clearly see in this cross section the spinal cord, and so they're showing you how the kidneys are kind of flanking either side of your spinal column. So renal ptosis is a condition in which one or both of the kidneys drop to a position lower than where they're supposed to be, and that can be because someone has lost a lot of weight very rapidly, and it's the fatty tissue that's around the kidney is lost, is burned, and their kidney drops. It can cause a ureter to get twisted and cause the urine to back up and exert pressure on the kidney tissue. Hydronephosis is the backup of urine from a urethral obstruction or other causes, and that can actually damage your kidneys very severely, which can lead to tissue death and renal failure. So looking internally on the inside of the internal kidney has three distinct regions. It has the cortex, which we've seen lots of times. Cortex means bark, or the outermost layer, and so that is the superficial renal region of the kidney. And then in the middle or near the center, we call it the renal medulla, which is deep to the cortex. And inside of that that medullary area, we have what are called the renal pyramids or the medullary pyramids. And they're called that because they actually are the shape of a pyramid. So the broad base of that pyramid is closest to the cortex. On the very tip of that, what you would think of as the top, so the top points internally. The tip of the pyramid, there's a little structure called the renal papilla. Um, and then the renal pyramids are separated. The space in between of them is invaginating cortical tissue that we call the renal columns. And so a lobe in the kidney is the medullary pyramid and the surrounding cortical tissue. And so each kidney has approximately 8 lobes. In the very, very center of the kidney, you have the renal pelvis. And coming out of that pelvis is a funnel-shaped tube, the ureter, that's going to drain the urine. When we look inside of the kidney, at the tip of each of those papilla is a structure called the minor calyce. It's basically a collecting area for the urine that's being formed. And then when 2 or more of those calyces join together, we call that a major calyce. And those all empty into the renal pelvis. So the flow of urine goes from the renal pyramid to the minor calyx to the major calyx to the renal pelvis out the ureter. And that's what we're talking about here. So if you take a look at the right-hand side, you can see what's called the capsule on the outside. Remember I said it's kind of a dark maroonish color or brownish color. And then what would look like the letter C, that's all the renal cortex. And then you have the area where you have those pyramids, and that area is called the medulla. So you can see the columns that are in between the actual pyramids. And then at the tip of each of those pyramids, you have a structure that's called the papilla of the pyramid. And that little collecting area just where that tip of that pyramid is draining into is called the minor calyce, and when they join together they form a major calyce, which ultimately forms the renal pelvis. So it's kind of like 2 little streams coming together to form a bigger river, which ultimately lead into the ocean. Pileitis is infection of the renal pelvis and the calyces, and pyelonephritis is infection or inflammation of the entire kidney. In females, this is usually caused by E. coli that's entered through the urinary tract. So very severe case of this can cause swelling of the kidney, abscess formation, and the renal pelvis fills with pus. And if it's left untreated, the kidney will become very damaged. This is something that's very easy to treat with antibiotics. So what about the blood and nerve supply to the kidney? So the kidneys cleanse the blood, and so they also adjust its composition. So it obviously has a very rich blood supply. So the renal arteries, if you remember, that came off the abdominal aorta, they deliver approximately 1/4 of the cardiac output to the kidney every minute. So within 4 minutes, the entire cardiac output has been cleaned. Or at least run through the kidneys. So the arterial flow for this is the renal artery, which leads to the segmental artery, which leads to the interlobar arteries, which lead to the arcuate arteries, and then the cortical radiate arteries. The old name for the cortical radiates are the interlobular. And then we're going to have a whole series of events that happens in the kidney. After the blood has gone through the filter in the kidney, it's going to start to return to the outside of the kidney, and it's going to do that via the cortical radiate vein to the arcuate vein to the interlobar vein, and then directly into the renal vein. So one unique thing, and I'll be pointing out a couple of other unique things, but one of the unique things about the blood flow through the kidney itself is that on the venous side there are no segmental veins. The nerve supply is via your sympathetic fibers from the renal plexus. So, taking a look at this picture, we're going to start at the renal artery that's out there over on the right-hand side. So find that renal artery, it's red, and as soon as that renal artery gets into the kidney and it's in the pelvis, it splits. And that splitting right there, those are called the segmental arteries. And then you'll notice if you follow one of those segmental arteries, they go up between the pyramids in that column area, and when they get in between those pyramids, they're called the interlobar arteries, because remember that a lobe is the cortical tissue on either side of the pyramid, and so interlobar, it's going in between the lobes. Then notice that that artery goes up and it goes over the top of the pyramid, and that's the arcuate artery. And then coming off of that arcuate artery are those red radiations into the cortex, and those are called the cortical radiate arteries. All right. Then let's look at the vein side, the venous side, following from going next to where those cortical radiate arteries are. Notice that there's a corresponding blue cortical radiate vein that goes down and becomes the arcuate vein that dips in between the pyramids and becomes the interlobar vein, and then it joins together and becomes the renal vein. There are no segmental veins. All right, so that's one of the unique differences. If you'd like to look at it from this part right here, you can follow what we were just talking about, going from the aorta to the renal vein, the segmental artery, the interlobar, the arcuate, then the cortical radiate. And then what we can't see because it's so tiny is that the cortical radiate becomes the afferent arteriole, which goes into the first capillary bed called the glomerulus, where all of the filtrate is going to be pushed out, or the plasma is going to be pushed out, and then coming out of the other side of that capillary is another arteriole called the efferent. Now there's something unique. Usually we see an arteriole, then a glomerulus or a capillary, and then we see a venule. So if you notice that you have a little box of dashed lines around that, what it's trying to show you is there's something unique going on there. So this is called a portal system. We've got an artery that leads to capillaries back into an artery that leads to another set of capillaries. And so those capillaries, depending if the type of nephron that we're talking about, is either going to be called a peritubular capillary or a vasorectal capillary. And then we lead into what we've already talked about, the cortical radiate vein to the arcuate vein, to the interlobar vein, to the renal vein. Notice that there was no segmental there, back to the inferior vena cava, and that blood is taken back up to the heart. Nephrons. Nephrons are the structural and functional units that form urine, and there are approximately 1 million nephrons per kidney. Each nephron has two main parts. It has what's called the renal corpuscle and then the actual renal tubule. So the two parts of the renal corpuscle. So the glomerulus is actually the blood supply. This is a capillary bed that's kind of all wound up into like a little circle, and it has lots of fenestrations or openings. If you remember back to blood vessels, fenestrations were large openings in that capillary. These are highly porous capillaries. They allow for efficient filtrate formation. And what is filtrate? Filtrate is the plasma that is pushed out of the blood that we're going to send through the nephron to be cleaned up. And then as much of it that we need, we're going to reabsorb back into our blood supply, and whatever is we want to get rid of as waste material, we're going to lose as urine. So the glomerular capsule is the first part of the actual nephron. So it used to be called the Bowman's capsule, and Bowman's capsule is a fine name for it. Just make sure you capitalize Bowman's. It is a cup-shaped structure that's surrounding the glomerulus. So if you want to think about it, it's kind of like a baseball glove over a baseball. The Bowman's capsule consists of the parietal layer, which is simple squamous epithelium, and the visceral layer that's actually clinging to the capillary itself, where it has branching epithelial podocytes. The extensions terminate in these little things called foot processes that cling to the basement membrane. The filtration slits between the foot processes allow for this filtrate formation to occur as it passes into the intercapillary space, intercapsular space. So taking a look at the picture on the bottom left-hand corner there, or the left-hand side down at the bottom. So you see where we are. Notice the red and the blue parallel vessel. All right, those are the arcuates. And then notice that the red vessel coming up, that one little piece going up, that's the cortical radiate. And then where it branches off and goes into that cup, that's called the afferent arteriole. And then you can see the glomerulus inside of there. And then the vessel coming out the other side is the efferent arteriole. Right? So we're looking at the Bowman's capsule, and what I really like about this slide is that it shows you over on the right hand side how the lining to the capsule changes as we move through the nephron, as we move through the tube. And we've seen this a lot. We saw this in the respiratory system, we saw this in the digestive system. As that the function changes of that tube, then we're going to see a change in the tissue, the epithelial tissue that's lining that tube. So there's the glomerular capsule itself. It is the parietal layer is going to be squamous, and then the part that's clinging to that little red vessel that you see over there on the top is the visceral layer. They show you the basement membrane, which is blue, and then these structures called podocytes that are clinging, or the end result of your parietal layer. They're turned into the visceral layer. The renal tubule or nephron is about 1.5 inches long. It consists of a single layer of epithelial cells, but each region, as I just mentioned, has its own unique histology and function. So there's 3 major parts to it. So you follow the tube from the renal corpuscle. So what the renal corpuscle is the Bowman's capsule with glomerulus inside of it, which represents the interface between the circulatory system and the urinary system. So there's 3 major parts to this tube. The part that's closest to the renal corpuscle is called the proximal convoluted tubule, and then it's going to make this loop, hairpin loop, called the nephron loop, and then you're going to have the distal convoluted tubule, which is the farthest away from the actual renal corpuscle in distance, but in actuality it's flipped over in front of it, and I'll show you that in just a second. That distal convoluted tubule is going to drain into a collecting duct, and the collecting ducts are going to be taking the urine that's been produced from lots of different nephrons. So it's not one collecting duct per nephron, but instead one collecting duct for hundreds of nephrons. And so the collecting ducts, when you look at the pyramids on the model, they kind of have a striped appearance to them. What you're seeing is the collection of all those collecting ducts coming together. So let's talk about your proximal convoluted tubule first. These are cuboidal cells with dense microvilli that form a brush border. This is going to increase the surface area in this portion of the nephron, and it also has lots of large mitochondria. This particular portion is all about reabsorption and secretion. This is confined to the cortical region of the kidney. So when we look at the first half of this tube, you have to kind of think backwards a little bit. So we're going to push the filtrate out at the glomerulus, and then we're going to try to keep whatever we want to keep by reabsorbing it off of that nephron back into the blood supply that is surrounding that nephron. So anything that stays in the tube is going to the toilet, if you want to think about it like that. So there is the proximal convoluted tubule. It has that apical side that's facing the lumen of the nephron, which has lots of microvilli. You see a lot of mitochondria in there because this is a very energetic portion of nephron. Then we have the nephron loop, also known as the loop of Henle. Again, notice the spelling of Henle. You have to use a capital H, and there isn't a Y at the end. This is a U-shaped structure, has 2 limbs. The portion coming off of the ascending, or the portion coming off the proximal convoluted tubule is called the descending limb. It is continuous with the proximal tubule, as I just said. The distal portion of the descending limb is called the thin limb because it has simple squamous epithelium. Then it makes the curve at the bottom and heads back up, and that portion is called the ascending limb. This side is thicker typically. In some nephrons it may be thin, but here's where we see that first change in the type of epithelial tissue that is lining that tubule. Here we'll have cuboidal or even columnar cells. And so here is the thin segment, or the downside of the descending side of the nephron. Then you're going to have the distal convoluted tubule. So it's going to be the last little squiggly line. This is going to be lined with cuboidal cells, but very few microvilli at this particular point. This portion of the tubule is functioning more in secretion than reabsorption. So secretion is actually putting fluid that managed to sneak back in to the blood supply, or something that didn't get out when it should have at the glomerulus, back into the tube for disposal. And the distal convoluted tubule is also confined to the cortex. So there is the distal convoluted tubule. Again, notice the lack of microvilli. So this picture might look familiar to you. We use this back in histology when we're talking about simple squamous and simple cuboidal. So what you're seeing, that thing in the very middle there that kind of looks like a big wad in the middle, that is actually a glomerulus. It's a wad of blood vessels, a capillary tuft, as they say. And then you see the glomerular space all the way around that. And then you see the squamous epithelial layer of the parietal layer of that glomerular capsule, which is that thin kind of purple line going all the way around. The collecting ducts. So remember, those are the tubes that are taking the newly formed urine off of the nephrons and taking it to the inner part of the kidney so it can be disposed of. So the collecting ducts, there's two types of cells that we find in those. One is called a principal cell. These have a very, there's not a whole lot of these, and they have very short microvilli. And these are all about water and sodium, water and salt balance. And so we'll be talking about those in the next chapter. And then you have intercalated cells. We saw intercalated discs in the heart. We're going to see intercalated cells. These are cuboidal cells that have a lot of microvilli, and there's actually two types of intercalated cells, A and B, both of which have to do with acid-base balance of your blood, which we will also be talking about in the next chapter. The collecting ducts, as I mentioned, receive filtrate from many different nephrons, and they run through those medullary pyramids, which is what gives those pyramids the striped appearance that they have on the models. All of the ducts fuse together at the very tip to deliver the urine through that papilla into the minor calyx. So there are your principal cells and your intercalated cells. So what's the difference? Well, they handle two different things. The principal cells have to do with water and salt balance, and the intercalated cells have to do with acid-base balance, or your pH. But you can see the difference in them as far as their microvilli. So there's the whole shebang back together again. So all the way back up there at the renal corpuscle, the Bowman's capsule, the glomerular capsule, the outer part of that. Then you see the first little squiggly line, which is called the proximal convoluted tubule. Then you see the descending limb of the loop of Henle where it goes down, and then you see where it curves and goes back up where it's called the ascending limb of the loop of Henle. And then you have your last little squiggly line, which is called the distal convoluted tubule, which goes into that larger structure called the collecting duct or collecting tubule. And notice all of those other places, those little stumps coming out, those are ports where other nephrons would be contributing their product as well. So let's talk about those nephrons for just a second. There are two major kinds of nephrons. Cortical nephrons, which make about 85% of the nephrons that you have. So remember that you have about 1 million in each kidney, so 850,000 of them in one kidney are called cortical nephrons, and they make up most of the cortex. Then you have 15% of your nephrons that are called juxtamedullary nephrons. These have a really extra long nephron loop that invades deeply into the medulla. The ascending limbs have thick and thin segments, and these are the ones that come into play when you're making really concentrated urine. In other words, you're not producing a lot of urine, you're saving most of your water. This is what you see when someone is not consuming enough water or they're dehydrated. Because there's so much time in the nephron loop, we can pull out more of the water, save more of the water, and lose less of it. So there's the difference between the two. The one over on the left-hand side is a cortical nephron. And that purple structure you see all around the outside of it, if you go back to looking at that blood flow through the kidney, that's that second set of capillaries called the peritubular capillaries that are picking up the water and any other substances that are being pushed off of that nephron that we're going to save and return to the blood supply. The one over on the right is a juxtamedullary nephron, and notice how long that one is. The purple structure you see on the outside of it is called the vasa recta, which is another form of a capillary bed, and that's the one that's pulling all of that extra water off and returning it to the blood supply, and thereby producing very concentrated urine. So again, here's the information on those nephron capillary beds. So the renal tubules have one of two types of capillary beds. They all have a glomerulus, right? And then the little short guys, the cortical ones, have the peritubular capillaries. The juxtamedullary nephrons have what's called the vasa recta. Your glomerulus, so a little bit of more detail about this. This is a capillary bed that is specialized in filtration. It's different from the other capillary beds that we've seen because remember we saw an arteriole going in and an arteriole going out. So the afferent enters into the glomerulus and leaves as the efferent. The afferent comes off of your cortical radiate arteries. The efferent either feeds into the peritubular capillaries if it's a short little nephron, or if it's one of the longer ones, then it feeds into the vasa recta. The blood pressure in the glomerulus is high. The afferent arterioles are larger in diameter than the efferents, and the arterioles are high resistance vessels. The peritubular capillaries, on the other hand, are low pressure porous capillaries that are all about sponging up or absorbing water and solutes that have been pushed out of the nephron that we want to keep. Again, remember, if it stays in the nephron, it's going to the toilet. So whatever it is that we want to keep, we push out of the nephron into these peritubular capillaries. These again come off that efferent arteriole, the exiting arteriole from the glomerulus, and they cling to the adjacent renal tubules in the cortex. These empty into the cortical radiate vein. The vasa recta was the long, thin-walled vessel that's parallel to the long nephron loops of those really long nephrons called the juxtamedullary nephrons. These come off the efferent arteriole that are serving the juxtamedullary nephrons, just like the peritubulars comes off the efferent arteriole. These are just in place. We call them this instead of peritubular capillaries. And they function in the formation, again, of concentrated urine. We have lots of time for that urine to travel through, or that filtrate to travel through that long nephron loop and pull off the water to save. So taking a look at some of these vessels in electron microscope, so you see what a peritubular capillary bed looks like, and the efferent and afferent arterioles that are feeding and draining them. There's that picture again, so you can start pointing out all of those. So one thing I want to point out right away is we look at that nephron like it's all stretched out and orderly, but you see that it's actually a great big old convoluted mess there. So when you're looking at the model, I always suggest that you start at the glomerulus and trace it with your finger, because they loop over each other. They get all twisted up, and sometimes it's hard to see if you're in the proximal convoluted tubule or if you're in the distal. So that leads us to talk about the structure called the juxtaglomerular complex, or the JGC. And so every nephron has one of these. And what it is the interface between the circulatory system and the nephron. So it involves modified portions of the distal portion of the ascending limb of the nephron loop, or some books say the distal convoluted tubule, and the afferent arteriole, rarely, but sometimes the efferent arteriole. And this is kind of like the quality control. This is really important for regulating the rate of filtrate formation. How much filtrate are we pushing out? And this is where the blood pressure component comes in. So there's three different cell populations that we're going to see in the JGC. There's going to be the macula densa cells, and these are tall cells closely packed together that are located in the ascending limb or in the distal convoluted tubule. Then you're going to have in those macula densa cells, there are chemoreceptors that are sensing the sodium concentration of the filtrate. So realize the filtrate at this point is about to go into that collecting duct, because we're at the very last portion of that. Then you have the granular cells, also known as the juxtaglomerular cells, or the JG cells. These are enlarged smooth muscle cells of the arteriole. So that afferent arteriole that's coming in, the cells on one side there are called the granular cells. And these have mechanoreceptors that are sensing blood pressure in the afferent arteriole as it's entering into the glomerulus. This is where that whole renin-angiotensin system comes into play. So if you remember how that system went, and I certainly hope that you did, remember that when the kidney detects low blood pressure, it causes the release of the enzyme renin. Well, this is where that happens. Then finally, what's in between those two sets of cells that I just talked about are called the extraglomerular mesangial cells. So they're located in between the arteriole cells, those granular cells, and the tubule cells, the macula densa, and they're interconnected with gap junctions. So they send signals back and forth between those macula densa cells and the granules. So here's what it looks like. So here, let me orient you. So look at the red tube coming in from the bottom. That's the afferent arteriole leading into the glomerulus. And then the efferent going out the other side. So notice on the kind of the part of the afferent arteriole going in on the left-hand side of that, closest to that little purple area, those are the granular cells right there that they're pointing out. Then you have that tube that you see in cross-section. That is going to be either the ascending limb of the nephron loop or the distal convoluted tubule, just depending on which book you're looking at. And notice the purple cells that they point out there, that they highlight there. Those are the macula densa cells. So the cells that are sitting in between in that purple area, those are the extraglomerular mesangial cells. So here's what happens. As blood comes in to the glomerulus, there's a little mechanoreceptor located up there by those granular cells, and if that blood pressure is too low, it's going to cause the release of renin, and then renin is going to facilitate that whole process of trying to raise the blood pressure. Different set of issues going on, completely different thing going on when we took a look at what the macula densa cells. The macula densa cells are determining how salty the urine is, how much sodium chloride is in the urine, and if we need to hold on to some of our water, it's going to cause us to reabsorb more of that sodium, so water follows it. And so those are chemoreceptors. All right, so two different things going on there. One has to do with the low blood pressure and the granular cells, and the other one has to do with the sodium in the urine, which ultimately is going to affect the blood pressure as well, but it's with the macula densa cells. Let's look a little bit at the physiology of the kidney. So roughly 180 liters of fluid are processed daily, but only about 1.5 liters of urine is formed. So think about that for just a second. That's an enormous amount of filtering, and we actually save most of what we filter. So your kidneys filter your entire plasma volume 60 times each day. They consume 20 to 25% of the oxygen that's used by the body at rest. The filtrate, which is what's produced by glomerular filtration, is basically your blood plasma minus the proteins. And urine is produced from that filtrate. So we produce urine, which is less than 1% of the original filtrate, which means that we reabsorb 99% of what's pushed out at the glomerulus into that nephron and conserve that. What's in the urine is metabolic waste and any unneeded substances. There's three processes that are involved in urine formation and adjustment of your blood composition. So the first thing is what happens at the glomerulus. They refer to that as glomerular filtration. This is what's producing the cell and protein-free filtrate. So there shouldn't be any cells in there, and there shouldn't be any protein in that filtrate that's being pushed out at the glomerulus into the Bowman's capsule. Then the filtrate passes through the nephron, you have what's called tubular reabsorption, which is what's selectively returning 99% of the substances that were pushed out in the filtrate to the blood in the renal tubules and collecting ducts. And then finally, tubular secretion, which selectively is moving substances from the blood to the filtrate in the renal tubules and collecting ducts. So somehow or another, something didn't come out at the glomerulus, or it passed passively back into the blood, and it has to be sent out. So tubular secretion is putting it from the blood into the tube. This picture, I think, is great for explaining those three processes. So you see up there where number 1 is, that's where glomerular filtration is occurring. 2 is going from the tube into the blood, and 3 is going from the blood into the tube. So let's look at them in depth. Glomerular filtration is a passive process. There is no metabolic energy required for this. It's depending on hydrostatic pressure that forces the fluids and the solutes through that filtration membrane, which is what we have at the glomerulus, into the glomerular capsule. There's no reabsorption into the capillaries of the glomerulus at this point. Everything is being pushed out. Let's talk about the filtration membrane. So this is the porous membrane that's between the blood and the interior glomerular capsule, which is allowing water and solutes that are smaller than a plasma protein to pass through. Typically cells like white blood cells, red blood cells, etc. can't pass through. There are 3 layers to this. So this is not any different than what we saw with the respiratory membrane. So this is the fenestrated endothelium of the glomerular capillary, its basement membrane, and the basement membrane of the podocytes, the foot processes of the podocytes with their filtration slits. The slit diaphragms are what help to repel any macromolecules that try to squeeze through. So here's what they're talking about here. So we're making what's called this filtration membrane. So we've got what's covering what's on the inside of the glomerulus, and then its basement membrane is epithelial cells on the inside of the glomerulus, and its basement membrane, and then this membrane, which is the is really the visceral layer of the membrane that's in there, that's sitting on top of the glomerulus, and it's made of simple squamous as well. So we have this membrane that things have to pass through. So they're showing you that in close up there, and the filtration slits and such that make it so that large molecules can't get through. There it is in electron micrograph. And just a picture here showing you what's happening. So we've got the plasma, which would be in the capillary side, is trying to push things through. So the plasma, so you see the simple squamous cells of the capillary, and the basement membrane is fused with the basement membrane of the foot processes of the podocytes, which are the visceral portion of that membrane. And so we create this a very effective filter that only allows certain things to move through. So macromolecules get stuck in the filtration membrane, and if that happens, there are glomerular mesangial cells. So we just saw extraglomerular mesangial cells, which were between the granular cells and then macula densa cells, but we also have another type of mesangial cells at this glomerulus that will engulf any macromolecules that try to sneak out. This filtration membrane is going to allow molecules that are smaller than 3 nanometers to pass through, so water, glucose, amino acids, and nitrogenous wastes. Plasma proteins should stay in the blood because they're large, and that helps to maintain the colloid osmotic pressure. This prevents the loss of all of the water to the capsular space, because if those plasma proteins pass through, the water would follow through as well. Proteins in the filtrate indicate that there's something wrong with the membrane. So let's talk about what is happening at that membrane and what the pressures are that are affecting the process of filtration, because remember, filtration is passive. So there is forces that are pushing the filtrate out, and that is going to be your hydrostatic pressure from the glomerular capillary, so the water pressure basically from your glomerular blood pressure. So this is the chief force that's pushing the water and the solutes, what we now call the filtrate, out of the blood and into the glomerulus, or into the nephron. The pressure here is quite high. It's 55 millimeters of mercury, which typically when we were looking at capillaries at the entry to the capillary, we were roughly around 26 millimeters of mercury. The reason is that efferent arterioles the efferent arteriole is a high resistance vessel with a smaller diameter compared to the afferent. So we have the water pressure pushing it out, but at the same time, we have something that's keeping it from being pushed out, or the inward pressures, the sucking, keeping it from going back out. These are the ones that are inhibiting the formation of filtrate. So the hydrostatic pressure in the capsular space, which is about 15 millimeters of mercury, and then the pulling of the proteins that are in the blood, which is about 30 millimeters of mercury. And so between the pushing and the pulling, we create what's called the net filtration pressure, or the NFP, which is the sum of those forces. So we had 55 millimeters of mercury that were pushing out, minus 45 millimeters of mercury that were opposing that, which led to a net outward force of about 10 millimeters of mercury, which is the pressure that's necessary for formation of filtrate to occur. So the main controllable factor determining this is what's called your GFR, or your glomerular filtration rate. So they're just showing you here what's pushing the water out and what's keeping the water from being pushed out. So the net filtration pressure ultimately, so the pushing out beats out the pulling in, and so we basically end up with about 10 millimeters of mercury, which is what's pushing that out. Your GFR, your glomerular filtration rate, is the volume of filtrate that's formed per minute by both kidneys, which is usually between 100 and 125 milliliters per minute. The GFR is directly proportional to your net filtration pressure. This is the primary pressure in your glomerular hydrostatic pressure that we talked about, the water pushing it out. The total surface area that's available for filtration, which is the glomerular mesangial cells, control this by contracting, and then the filtration membrane permeability, which is much more permeable than other capillaries. A constant GFR is important. It allows the kidneys to make filtrate and maintain extracellular homeostasis. So this is the goal of the local intrinsic controls at the kidney itself, what they call renal autoregulation. Its job is to maintain the GFR at the kidney. So your GFR affects systemic blood pressure. It's an increased GFR causes the increase of the amount of urine that is produced, which lowers your blood pressure, and vice versa. So if you have too much water in your blood, you increase your GFR, and you push out more water from your urine, and you lower your blood pressure. The goal of extrinsic control is to maintain systemic blood pressure. So this is what the nervous system and the endocrine mechanisms are all about. So let's talk a little bit about this intrinsic control of renal autoregulation. This maintains a nearly constant GFR when the MAP is anywhere between 80 to 180 millimeters of mercury. So that is a pretty astounding thing. It kind of doesn't really matter what your blood pressure is. As long as your kidneys are healthy, then your GFR stays about the same. So there's a huge range there, that as long as you're within that range, your kidneys still do the job that they're supposed to do. Autoregulation ceases if you're out of that range. So there's two types of renal autoregulation: myogenic, which is always about muscle, and tubular glomerular feedback mechanism. The myogenic mechanism, this is the local smooth muscle that contracts when it's stretched. So if you have an increase in your blood pressure, that causes the muscle in the wall of the vessel to stretch, which leads to a constriction of the afferent arterioles, which is going to restrict the blood flow into the glomerulus and protect the glomeruli from damaging high blood pressure. This is how people with high blood pressure lose their kidney function, when it goes unchecked. So if you don't check your blood pressure and keep it in its normal range, if that blood pressure comes up to the glomerulus too high, it literally blows out that glomerulus. Decreased blood pressure causes dilation of the afferent arterioles, and so those two things together help to maintain that normal GFR that I was talking about, despite the constant fluctuations in your blood pressure. Then you have the tubular glomerular feedback mechanism. This is flow-dependent mechanism that's directed by those macula densa cells that were found in the distal convoluted tubule, or the last portion of the ascending limb of the loop of Henle. This is responding to the filtrate that's been produced sodium chloride concentration. So if your glomerular filtration rate increases, the filtrate flow rate increases, which leads to decreased reabsorption time as that filtrate passes through the nephron, which causes high sodium in the filtrate. The feedback mechanism causes constriction up at the afferent arteriole, which lowers the net filtration pressure and the glomerular filtration rate, which allows more time for that sodium to be reabsorbed. And of course, the opposite mechanism would be in play for a decrease in the GFR. So that whole juxtaglomerular apparatus is coming into play here. What about the extrinsic controls? This is the neural and hormonal mechanisms. The purpose of extrinsic control is to regulate the GFR to maintain your systemic blood pressure. Extrinsic controls will override the renal intrinsic control if your blood volume needs to be increased. When you're in sympathetic nervous system, this is under normal conditions at rest, the renal blood vessels are dilated. The renal autoregulatory mechanisms prevail. Under abnormal conditions, such as extremely low extracellular fluid or low blood pressure or low blood volume, norepinephrine is secreted by the sympathetic nervous system, and epinephrine is released by those adrenal medulla, the adrenal medulla and the adrenal glands, which causes systemic vasoconstriction, which will increase the blood pressure, constriction of the afferent arterioles, which decreases the GFR, and then it causes your blood volume and your blood pressure to increase. Then there's the renin-angiotensin-aldosterone mechanism. So for the umpteenth time this semester, we need to make sure that we understand this. This is the main mechanism for increasing our blood pressure. There are three pathways to renin release by those granular cells in the afferent arteriole: direct stimulation by the sympathetic nervous system for fight or flight, stimulation by activated macula densa cells when the filtrate sodium chloride concentration is too low, or reduced stretch of the granular cells. So there it is once again. So if you haven't gotten this by now, please, please, please make sure that you come and see me immediately. You absolutely have to understand this. So just as a reminder, either sympathetic stimulation, hypotension, or too little sodium in the filtrate, because the filtrate's moving through the nephron too quickly. At the kidney, at that afferent arteriole coming in, is going to lead to the release of renin, which is going to come in contact with angiotensinogen and convert it from the inactive form of angiotensinogen into angiotensin 1. The angiotensin 1 is going to float through the lungs eventually and come in contact with ACE, which is angiotensin converting enzyme and convert A1 into angiotensin 2. And then angiotensin 2 is going to work on all those different systems to bring the blood pressure up. It's going to work on the adrenal cortex, cause it to release aldosterone, which causes us to hold on to sodium and retain more water, thereby increasing our blood volume and therefore increasing our blood pressure. It's going to work on the pituitary gland, cause the release of antidiuretic hormones so we don't release as much urine, thereby holding on to more sodium, holding on to more fluid, increasing our blood volume, and increasing our blood pressure. It's going to work on the hypothalamus and cause you to be thirsty, which will increase your blood volume, thereby increasing your blood pressure, and so on. You can work your way around that whole thing. So again, the idea is always that we are increasing the blood pressure with this system. There it is in a different form. If you like this version better, I have a hard time seeing this, but some people see it this way, and this is perfectly fine if you get it this way as well. So there are some other factors that affect your GFR. The renal cells release a variety of different chemicals. Some of those chemicals can act as paracrines, which are local chemicals that affect the renal arterioles. Some examples of those chemicals are adenosine and prostaglandin E2. Some cells make their own locally acting angiotensin 2. This reinforces the effects of the hormonal angiotensin 2. And then finally for this section, anuria, this is abnormally low urine output, meaning less than 50 milliliters per day. This might indicate that the glomerular blood pressure is too low to cause filtration. Renal failure and anuria can also result from situations in which the nephrons have stopped functioning in cases of acute nephritis, transfusion reactions, or crush injuries. Okay, that's the end of part A. I'll see you for part B.